Welcome everyone. So as I discussed in my previous video regarding the protoplast 1 protoplast 2 and formation of the somatic hybrid, now it is very important for us to identify the hybrids. So when we have a mixture of parent 1, parent 2 as well as somatic hybrids, it would be difficult for us to differentiate whether the protoplasts are fused to produce somatic hybrids, yes or no. Now in a simple way, if at all I have to tell you, in a beaker of this mixture, I will be having protoplast 1, I will be having protoplast 2, and I also have somatic hybrids here. As we all know, the protoplasts are quite small, which we cannot see through our naked eyes. So how can we differentiate between P1, P2 as well as somatic hybrids? For this we have a separate technique called as identification of somatic hybrids. Okay. So in identification of hybrids, what we try to do, we either try to check somatic hybrids and remove the rest of the cells, or we try to check the protoplasts, try to take out the somatic hybrids. So this can be done in many different ways. Some of the important techniques are staining method, centrifuge, and the complementation technique. When we discuss about staining, centrifuge as well as complementation technique, out of these three things we would go for complementation technique. I'll tell you what exactly is the reason. When it comes to matter of your staining technique, I try to stain the protoplasts. Either parent 1, parent 2 try to take the protoplast, or somatic hybrid could try to take the protoplast. So once identification is done, okay, it's only the identification what we do. After the identification, we also have to extract the protoplast. So which is a different, or which is a task, or an additional task basically. So I would stain with carbon, fusion, or fusion is what we basically call the stain as. So this stain or the staining technique will try to stain only the somatic hybrids, whereas P1, P2 are just left behind. So whichever the stain cells, have to take it, have to extract it, and have to go for my further process. So that is one thing. When it comes to matter of your centrifugation, as we already know that when it comes to matter of P1, P2 as well as somatic hybrids are quite heavy because they have two nucleus, they have two cytoplasms. So when it comes to matter of your centrifugation, P1, P2 will be at the top, that is somewhere like a supernatant. Somatic hybrids would come to the lower portion when it comes to matter of the centrifuge or density gradient. So in that way we try to separate out the normal protoplast with that of the somatic hybrids. But the important technique starts now, where we have something called as complementation technique or complementation method. Complementation method, it's a process where some genes of P1, some genes of P2 would try to complement to produce a new variety, and this new character would be used as a detection purpose. The new character would be used as a detection purpose, for which we have some of the examples. Let me write the examples now. First one is antibiotic complementation technique. So let me write you on the examples. Petunia, So what exactly is in this example is, I have a particular plant variety called as Petunia hybrida, and I also have a plant variety which is called as Petunia parodi. So now what exactly is happening here is, if I try to take the protoplast of plant 1 and protoplast of plant 2 and try to grow protoplast of this separately on an actinomycin media, this would be dead, this would be dead. Whereas the fused form of P1, P2, where you try to see the fused one, if I try to grow, they survive. So the reason here is, one of the gene from P1, another gene from P2 would try to complement so that a new gene is produced or a new character is produced, and the new character is actinomycin. Okay, in this particular actinomycin, the plant would survive. The next example what you have is Nicotiana tabacum, that is nitrate complementation. I have plant 1, I have plant 2. Let me erase this off. So I have plant 1 and plant 2, CNX minus, NIX minus. Molybdenum. Okay. So the explanation is almost similar to the first one. Nicotiana tabacum, which is a plant, in this nitrate complementation is a technique what we try to do. The plant 1 or the protoplast 1 having CNX minus, that is CNX is a gene which codes for molybdenum, whereas the plant 2 has NIX gene which codes for NR-apo enzyme. If I try to grow plant 1 in nitrate medium, so media which consists of nitrate, plant 1 cannot grow. Same way, when I try to grow plant 2 in the same nitrate medium, plants cannot grow. But if at all I try to fuse P1, P2 to get a plant variety, a cell variety of P3, they can grow in nitrate media. And here, molybdenum as well as NR-apo enzyme, these both would be combined to form a complete enzyme. This complete enzyme would complement the newly produced P3 and they would survive in the nitrate media. So this is another important technique what we go for complementation method to differentiate between the normal protoplast as well as, I've already erased, the somatic hybrids.